Hi everyone, welcome back to the Bioinformatics Coach channel. In today's video, I am going to give you 4 software you need to have in your bioinformatics toolkits. So let's get started. The first software is Linux. Linux is a Unix-like operating system, just like macOS. There are several distros available. We have Ubuntu, we have CentOS, we have Fedora, etc. And the reason why Linux should be on the list is that Linux is free. You don't need to pay a dime in order to use most of the Linux distros. Okay. Linux is open source, so you can always download, modify the codes free of charge. Okay, so that's one of the reasons why you have to use Linux. The other reason is that most bioinformatics software, especially those that process the raw data, work only on Unix-like operating systems, i.e. Linux and macOS. Okay, and since Linux is free, it makes sense for you to use Linux as one of your software for analyzing bioinformatics data. The other reason is that Linux is able to handle computationally expensive tasks. Okay, and that is why these softwares also work on only Unix-like operating systems, because the data sets, especially the raw ones, when processing can take a longer time, and then also they are heavy. The files are huge and they are complex in some cases, and therefore processing them becomes computationally expensive. And that is where Linux comes in, because it's able to handle this kind of pressure. The other reason is that Linux is stable. Linux rarely crashes. Okay, and that is why it's also able to handle computationally expensive data, because if you're analyzing bioinformatics data, especially the raw data, it can take days, weeks, or maybe longer. Okay, and therefore you need an operating system that will be stable, that can withstand this pressure, so that you can analyze the data. That is where Linux comes in. So Linux is very, very important. Okay, it's one of the tools that you should definitely have on your list. The other reason is that Linux has a powerful and flexible scripting environment. Linux allows you to combine commands in almost an unlimited fashion. Okay, Linux also has tools and utilities, and that helps you to process text files. Most bioinformatics data sets, almost all the bioinformatics data sets, irrespective of the format or file extension, are fundamentally text files. Okay, and so if Linux has all these utilities to help process text files, then it makes sense to use Linux to also process our bioinformatics data. So that's about it. Linux also has a multi-tasking and a multi-user environments. Okay, so Linux allows you to perform multiple tasks, okay, on the same system, in some cases at the same time. There will be no issue, because it has that property. Okay, again, this also ties into the stability. Okay, so that's about it. Linux also allows the multi-user environments, meaning that several users or multiple users can log into the system at the same time, and each user can perform his or her tasks, okay, without it interfering with other users' tasks and activities. So that is why when you have a computer cluster, you can see that the cluster is able to allow multiple users to log in and then use the system, and there will be no issues, because Linux has that property. So that's about it. Now let's move on to the next software, which is Anaconda. Anaconda is a software package manager, and so it allows you to install and uninstall software. One great thing about Anaconda is that it automatically installs all dependencies of the software that you want to install. All that you have to do is to issue your command or commands to install the software of interest, and then Conda will do the rest. Okay, Conda will automatically install all dependencies. So Conda will, in other words, do all the heavy lifting for you when it comes to software installation. Okay, or let's say software management as a whole. Anaconda also allows you to install different versions of a software on the same computer via the environment system. Okay, So with Anaconda, you use the Conda commands, okay, to create environments, and then in the environment you will install your software. So if you want to use a software, you just have to activate the environment, and once you are done, you can deactivate the environment. And for those who are struggling to install Anaconda in a Linux environment, I have a tutorial that shows you how to install Anaconda in a Linux, so you can just watch that tutorial to set up your PC. I also have another tutorial that shows you how to use Anaconda to install bioinformatics software, so you can also watch that video. I'll leave all these videos in the description box for you to check them out. Anaconda also allows you to install R and Python. R and Python are heavily used in the field of bioinformatics, and therefore Anaconda being able to do this installation is a great thing for us. So that's about it. Now let's move on to the next software, which is R. R is a programming language. It's also a statistical software. Okay, so R is heavily used in the field of bioinformatics. You can use it for downloading data. You can use it for data analysis. You can use it to manipulate data, etc. Okay, R also allows you to perform statistical tests. It also allows you to generate plots and figures. As a matter of fact, the plots and figures that R generates are publication quality, meaning that you can use those figures in your papers that you want to publish, because R has lots of packages for data visualization. So it's a great tool, okay, for analysis and also for visualization. And there are situations where you may not be able to escape from R. Okay, if let's say you want to perform gene expression analysis, which by the way I have covered some tutorials on, I'll leave those videos in the description box. If you want to perform gene expression analysis, let's say you want to use ionistic data, you will need to first get your FASTQ reads. You need to perform QC. You need to map the reads to a reference genome. You need to generate a read count, normalize, and then you identify the differential express genes. The packages available, most of the packages available for identify differential express genes work only in R. That means you can only use them via R. Okay, talk about HR, talk about DSIC2, talk about Borgon, etc. All these packages are available in R. Okay, so in such a situation, you have no option but to use R. So take note of that. So R is a great tool, and you should definitely have it on your software list. There are other analysis that R can be used for, such as population genomics. You want to generate Manhattan plots, you can use R. You want to generate PCA, you can use R. You want to analyze VCF files, SNP data in VCF formats, you can use R. There are lots of packages to help you perform bioinformatics tasks. So R is a go-to programming language when it comes to bioinformatics. Now let's move on to the next software, which is Python. Python is a general purpose programming language. It has lots of packages for bioinformatics tasks and also data science as a whole. Okay, and so with Python, there are packages to help you analyze your data. There are packages to help you to download data. There are packages to help you to manipulate your data. There are packages to help you to visualize your data. So I mean, there are lots of packages. Okay, so what you have to do is to read the documentation and then look at what works best for you. There's this question that often comes up: Python, which of them should I use for bioinformatics analysis? I have already covered a talk on that, but I'll just give a brief statement here. Python and an R. If you want to select one, it should be based on what you want to achieve at the end of the day. Both languages are great, but then if you know what you want to achieve, then it's easy for you to get documents that will tell you whether there are packages in R or Python to help you achieve that. So that's something you have to consider. Okay, so both languages are great. Now with Python, it also has its strengths its advantages. Okay, let's just say you want to perform some machine learning tasks. You want to develop predictive models, okay, or you want to apply some machine learning models on your data, okay, on your bioinformatics data. Then Python is what I recommend because Python has lots of packages for machine learning. Okay, it's heavily used in the field of machine learning, and therefore it's likely that you have documents and tutorials and materials to help you um, um, apply the machine learning models on your data. So that's about it. So if it comes to machine learning, then I'll highly recommend you use Python. So that's about it. So if you're also a biologist and you want to learn Python programming, I have a separate playlist that shows you how to do that. I have two playlists, one for beginners, one for intermediates. So you just need to look at your level and then decide which of them is good for you. But all these tutorials are reproducible and all the codes are also available. So just go and then check them out. The videos, I'll leave the playlist to those videos in the description box for you to just use them. So with those videos, I try to explain the concepts of programming using biological data types, using biological processes. So it will be good for you, my viewers who are hardcore biologists. So in fact, when it comes to Python, there are some tools that also use Python in the background, like GATK, for example, uses some Python scripts when performing variant calling. I think HiSat2, this tool is used for mapping RNA sequence to a reference genome. It also uses Python in the background to do the mapping. So that's about it. So in a nutshell, these are the software that, in my opinion, you should have on your software list because you will definitely need some or all of them and when you want to analyze bioinformatics data. Let me also get your comments. If there are other software that you think should be included, just put them in the comment section and let's have some discussion. So that'll be all for this tutorial. Thanks for watching and I'll see you in the next session. Goodbye.